Thank you for accepting the invitation to participate in the lecture in this forum. Can you hear me?
Yes, I can.
Many of our Chinese followers are concerned about the effect of NR on longevity. Now, if you're ready, shall we start?
We should start. Thank you very much. Really pleased to be able to join you today. I hope everyone is staying safe. Hearing some scary things about the COVID outbreaks in northern and southern China these days. I'm hoping everyone is staying safe. So I'd like to give my talk in sort of two parts, boosting resilience with nicotinamide riboside and busting longevity myths with analytical reasoning. And my next slide has my disclosures. So as you may know, I developed the intellectual property on uses of nicotinamide riboside, which were developed by ChromaDex, and I advised ChromaDex chief advisor, and I'm a chief advisor of a company called Juvenon, which is also in the NAD space. I'm a co-founder of an NAD company. You're looking now in the north view from my laboratory at City of Hope at the San Gabriel Mountains in the Los Angeles County in California. Now, I gave this talk just a week ago in Las Vegas, Nevada. I don't know if you folks know about Las Vegas, Nevada, but it's the main gambling city in the United States. And it was a longevity conference. And I told everybody that attending a longevity conference is a little bit like coming to Las Vegas, in the sense that there are professionals that, will use people's greed and fear or hopes and anxieties to basically part you from your money. And so I want to ask the question, whether it's any different now. And one of the problems with longevity claims is that throughout the entire history of Western civilization, and may be true in Chinese history as well, there have been legends. So there was a legendary fountain of youth that was written by Herodotus. Herodotus is considered the founder of history in the Greek tradition, and he claimed that there were waters in a mystical place and people could bathe in these waters and never grow old. And of course, he was never able to identify where the fountain of youth was, what was in the waters, if those were real people, but it was a legend. And these legends have been carried out over the centuries by many, many people, including Ponce de Leon, who is one of the Spanish explorers that came to the United States. So let me, just to bring it up to the present day, let me read you a letter that I received on March 25th. It's impossible for me to remain silent when I consider the revolutionary progress we have made in understanding the aging process. As a friend of such and such a company, I thought I would send a newsletter now and then to update you on the spectacular events unfolding. In summary, we absolutely have within our hands the technology to manipulate and reverse aging in every tissue and system. Let me put this another way. We can take senescent cells and make them immortal by manipulating certain aging genes, make them senescent again at our caprice. This demonstrates that we have found the genes that regulate the aging process. Furthermore, we've demonstrated that these same aging genes are directly involved in the generation of a vast array of age-related diseases, including Alzheimer's, atherosclerosis, geriatric hypertension, skin aging, wrinkling and balding, osteoarthritis and osteoporosis, just to name a few. The biggest surprise was the Alzheimer's connection. The cells of the central nervous system operate by a lot of different rules than seen in the other tissues of the body. I don't think the most optimistic of us could have predicted that the brain would age in such a similar way as the connective tissues. This powerful technology now needs to be engineered into a form that can be administered to people. All of this testing will certainly take the better part of the decade, but I'm impressed by the enthusiasm of 3 major pharmaceutical firms that have contacted me to take on the work of bringing this technology to market. And now I'm going to ask all of the members of the conference to guess who said this and when. Do you think this was written in a letter 9 months ago by Rick Klausner at Altos Laboratories? Or do you think it was 5 years and 9 months ago by Mark Allen of Elevian about, GDF11? Do you think that it was 9 years and 9 months ago, Craig Venter? Or do you think that maybe it was more like 18 years and 9 months ago, said by David Sinclair at Sirtuins? Any guesses? The actual answer is this was claimed over 32 years ago by Michael West at Geron. And you can't say that he didn't succeed because we don't know how to do an aging clinical trial. Like, how would you conduct an aging clinical trial? Enroll people that are 105 years old and see whether you're extending lifespan in a placebo-controlled trial? Theoretically, you could do that, but people that are 105 are not typical of the general population. No, West claimed that these molecules could be tested in atherosclerosis, skin aging, wrinkling, any number of clinical trials that would be easy to do. So what I'm emphasizing here is that for thousands of years, people have been claiming fountains of youth. For decades, biotech scientists in academia and in industry have been claiming breakthrough results in longevity that have not been delivered. So that's a real warning to this audience. And in fact, the best-selling book of all time about aging has this as a subtitle: What if aging is a disease and that disease is treatable? And I'm showing you a park that was built in the early 20th century by a woman named Louisa Day McConnell in Florida, where she claimed that Ponce de Leon found the Fountain of Youth in 1513, and she basically built a sign over a water well on her property, and she sold this water to people as an elixir of life. And as you can all imagine, all of the people that bought that water and tasted that water 100 years ago are dead. This book that has the subtitle, What if Aging is a Disease and that disease is treatable, has promulgated 3 completely false ideas. The first idea that's false is that there's a class of genes called Sirtuins that are dominantly acting monogenic longevity genes that are conserved from yeast to humans. I'll show you during this talk that that is not true. The book further claims that Sirtuins are activated by compounds identified in stressed plants like resveratrol. That's not true. And third, the book makes claims that we can rejuvenate tissues in mice and that people are, quote, aging in reverse. And I'll show you the problematic nature of those types of claims. So I recently published a major review article. I think it has about 130 or more citations in it, entitled Sirtuins Are Not Conserved Longevity Genes. I give you the URL for this paper right here. And now the original result from the late 1990s in the yeast system, yeast Saccharomyces cerevisiae, was that a gene called SIR2 extends lifespan in one type of yeast model of lifespan. That's true. That is a fact. But as I show in this publication, it's only meaningfully extending the lifespan of 1 out of 5 million 1 out of 5 million of the cells in that culture. Only the very oldest, what are called yeast mother cells in the culture have the beneficial effect of SIR2 on lifespan extension. SIR2 actually shortens the lifespan of every cell in the other type of model of yeast aging, which is called chronological lifespan. That was shown in a paper by Walter Longo in 2005 and published in Cell. In 2001 and 2004, it was claimed that extra copies of the SIR2 gene extend lifespan in worms and in flies. Turns out, by the year 2011, there's another paper in Nature showing neither of those results are reproducible. So it turns out that there was an artifact of strain background in the worm system and in the fly system. When you properly do the controls, extra copies of SIR2 related genes do not extend lifespan in simple animals like worms and flies. And in fact, in 2016, it was shown that loss of one copy of the Drosophila SIRT gene extends lifespan in flies in response to calorie restriction. Not extra copies, but loss of one copy. And the problem is that there are thousands of citations to these papers that are incorrect and irreproducible. And there are literally only about 10 or 15 citations to the paper that shows no, actually the opposite of that is true. But nonetheless, 5 different groups went ahead and made overexpression models in mice. 4 groups did a whole body SIRT1 overexpression. None of those models showed extended lifespan. There's one group that did brain-specific overexpression of SIRT1, and they reported 2 different transgenic lines that overexpressed SIRT1 in the brain. One of them showed no lifespan extension. The other one did, was presumably some type of a position effect. So effectively, in no model of overexpression of SIRT1 in a mouse, fly, or worm does SIRT1 extend lifespan. And then the narrative was changed somewhat, and people said, oh, no, really, we should be talking about a gene called SIRT6. And in a sex-specific and strain-specific manner, there are some lifespan extension results with SIRT6, but fundamentally, SIRT6 is a positive regulator of gluconeogenesis. And as mice age, their blood glucose goes down, so that if you improve their gluconeogenesis, they're healthier. But you don't want to do that in a human being, because as we age, our blood glucose tends to go up, as you heard from the last talk, and that's really not good for us. So that's basically bad in people. And the problem, as I said, is that there's inaccurate information in hundreds of review articles with hundreds to thousands of citations. There's thousands of research projects that have been mispremised. I'm sure every person in this conference has read papers that were premised on the basis of SIR2 genes being conserved as longevity genes. And that creates a framing bias and a confirmation bias that is accompanied by hype in the media, and then something called harking, where you make a hypothesis after the results are known. And that's just not really the right way to do science. We need to have falsifiable hypotheses. And if SIRT overexpression is supposed to extend lifespan, we can't be going around and making excuses for why it didn't work and why something really should be interpreted as a positive result. So billions of dollars have been misallocated on the basis of this. And the implications are rather profound, because if there were dominantly acting monogenic, high penetrance longevity genes, then CRISPR would be great, right? Because we would all want to CRISPR in extra copies of Sirt1 or whatever the dominantly acting longevity genes are, or supplements that activate Sirt1 would make sense, because like resveratrol would make sense, because if Sirt1 extended your lifespan and if resveratrol activated Sirt1, and neither of those things are true, then that would be an amazing story. And if all it is really is a story, it's driven by skilled storytelling and not data. And so let's do what we call real talk of the genetics of animal lifespans. So all living things have characteristic lifespans that are genetically encoded and environmentally modifiable. So in the bottom of the slide, I have pictures of a mouse and a naked mole rat. They're about the same size, and they differ in their lifespan, even though they have basically the same body plan, the same basic genomic architecture, but the naked mole rat lives about 10 times longer than the mouse. Now, we know that lifespan is easy to shorten environmentally. It's not so straightforward to extend lifespan. And so, for example, we don't have a gene set that allows us to survive at 50 degrees C, right? We're susceptible to death by virus, death by gunshot. We know that overnutrition shortens lifespan by multiple mechanisms. Smoking shortens lifespan. So let's do a thought experiment now. And let's do a thought experiment about a very simple bacterium with, let's say, 1,000 genes. And this bacterium grows very well at 45 degrees, and you can get it to grow and survive at 49. But it doesn't grow at 50. Now, if we apply a selection to this bacterium, how many mutations do you think that you would need to get this bacterium to go from growing at 49 to growing at 50? Now, I don't know the answer to this thought experiment for certain. It's just a thought experiment. But I would argue to you that if there are, let's say, 400 or 500 essential genes out of 1,000 genes in this bacterium, we might need as many as 400 or 500 mutations, because none of the proteins in that bacterium have evolved or have been selected to function at 50 degrees. So you're going to need mutations in DNA polymerase, RNA polymerases, repair proteins, proteases, ligases, everything. And that is a very important concept when you think about the genetics of something as complex as longevity. It was rather absurd to think that there was a gene that was found in yeast, which yeast existed prior to the emergence of any animal, that some gene in yeast would anticipate the causes of death in animals and be limiting for the survival of all types of animals. That was a rather remarkable idea. In fact, what we know about animal evolution is that the things that are under selective pressure are the ability to acquire food, right? Babies are hatched, or, eggs open. The animals have to be able to get to the ability to acquire food, avoid predation, get to sexual maturity, and then either fecundity, which means lots of babies, or caretaking of the babies until the offspring can do all of the above: get food, avoid predation, and become sexually mature. And with multiple cycles of reproduction and caretaking, then you get some longevity. But basically, longevity is not a directly selected trait. Animal evolution doesn't really select for the ability to outlive our reproductive capacity by more time than required to provide reproductive advantages. And this was basically established and put in a book by 1991 by Michael Rose and other people that did experimental work and theoretical work that is foundational for understanding the genetic basis of lifespan in animals. And if you think of the longest lived animals, like bowhead sharks and certain types of whales and turtles that live over 100 years, if a shark lives for 180 years, it's capable of reproducing all of that time, because that's the selective pressure is basically operating on the ability to pass on the gene pool. And that means, and pardon me for the X-rated picture here, but the implications are that the brain, the musculoskeletal, the circulatory, respiratory, and digestive systems get the mileage they get. They get the longevity that they have to support reproduction. And that longevity is not itself under direct selective pressure. Successful reproduction over time integrates all of the other fitness measures of having a brain and having mobility and having sex appeal, basically, as an animal, and surviving winters and surviving summers and surviving drought and so forth. And so longevity is an emergent property. And so longevity is encoded by virtually all of our genes, not like single dominantly acting genes. And so if there aren't single dominantly acting genes, then CRISPR is an amazing technology, but CRISPR doesn't actually enable us to simply engineer ourselves to longer lifespan. So the general rule is that reproductive capacity in animals approximates animal lifespan, but there's a very, very important exception to that rule, and that is the human female. The human female spends much of her life, 30, 40 years, past the point of reproductive capability because of menopause. Average onset of menopause is the age of 51. And as you know, women have life expectancy into the mid-80s. And we understand that because follicular atresia, if you look at these rates, is really rapid. This is a log scale of the number of germ cells and follicles. There's 1.5 million follicles when a baby girl human is born. There's only 300,000 left by the time she reaches puberty. She's never released a single oocyte for maturation, and she's already lost 80% of her primordial follicles. And if this were simple entropic decay in a log linear plot, then this would be a line. But you can see that follicular atresia is constantly accelerating, which means aging is a program. We know that aging is a program for women's reproductive lifespan, and we know that aging is a program in general. And to prove that genetically, if you go around longevity researchers and you ask them, what is the most convincing monogenic longevity genes? No one will give you an example of a dominantly acting gene. I'd be shocked if anybody gives you an example of a dominantly acting gene, but everyone will give you examples of loss of function genes in the growth hormone pathway. So like growth hormone, IGF-1, the receptors, the transcription factors in the pituitary. So loss of function of these genes give you the Ames dwarf mouse, the Snell mouse, the little mouse. These are all tiny little dwarf animals. They are infertile. They're incapable of surviving on their own. And so basically these genes are conserved for growth. Loss of both copies gives you extended lifespan, but the genes that encode growth hormone and growth hormone receptor, when lost, will give you extended lifespan. So this is not something that you can easily CRISPR in. In fact, if you were to walk around that longevity conference I attended a week ago, there are people selling things like steroids and human growth hormone to kind of enhance vitality, but they will probably, based upon genetics, shorten human lifespan. Now, what about the claims in a popular book of epigenetic resetting, that we've gotten mice to age in reverse, and that we get people to age in reverse? Well, obviously, what we're talking about here is the use of Yamanaka factors. There was a paper, there was a study that was described in a book by David Sinclair, in which he described an experiment that he was doing. And then about 4 years later, he published the actual paper in Nature in which he claimed to, get this result. But the thing is, we know that Yamanaka factors are oncogenes. We know that there's a very low efficiency of generating any one lineage, and that they're a teratoma hazard. And when we use Yamanaka factors in ex vivo, in tissue culture, we know that you can put them into a fibroblast, and you can generate induced pluripotent stem cells at a low frequency, but you're also generating cells that die, you're generating cells that form teratomas, malignancies, and so forth. So you're cloning things out in vitro. It's an entirely different thing when you do it in an animal. And if you read the methods, it says that the investigators were not blinded to outcome assessment. So they were basically biased by, I believe, their boss telling the entire world what the result was going to be. Now you might say, well, look, I've seen experiments published in whole mice by Belmonte and others in early 2022, where they did all 4 Yamanaka factors in a doxycycline induction system, and they claimed that they, reversed the epigenetic age of these mice. But there's no real functional test. There's only 21 mice in the minus dox group and 15 mice in the plus dox group. And you can see that at 22 months, the survivorship is only about 80%. So these mice with doxycycline are dying, just like the mice without doxycycline. The paper said they didn't see any cancer. Well, we all know that means they weren't looking for any cancer, because 50% of black 6 mice, if you let them go out to their natural cause of death, are going to die from cancer. So if you were going to do this experiment properly, you would have to have something like 1,000 mice in each arm, intervention and control, and you'd have to give those mice to a third party who's not invested in what the outcome was, and have them look very carefully at what the survivorship was and what the health and the fitness and the tumor incidence was in those mice. So we cannot say, based upon epigenetic age, that we know how to in vivo reprogram mice, let alone humans. But there are people that have claimed age reversal in humans. And this is what I call the tautology problem of biomarker-informed longevity trials. So if you want an example of a tautology, it's a statement like this: if it's in stock, we have it. It's not, a meaningful statement. So the hypothesis is that there are these aging clocks, and that, for example, DNA methylation biomarkers are modifiable risk factors for mortality. That's an idea, that's a hypothesis. There's not overwhelming evidence in favor of that hypothesis. Then there's a second hypothesis that is stacked on top of that, which is that intervention X will lower the risk for mortality as measured by DNA methylation. So this is where someone, it was Greg Fahey and others, gave human beings, a small group of people without placebo control, a cocktail of drugs, including human growth hormone, which we know from genetics is likely to accelerate aging, plus DHEA, which is a steroid precursor, plus metformin. And then the finding was their methylation age was lower at the end of this intervention than before. And the headline result was older people grow 2.5 years younger. This is not a valid interpretation. That is just saying that you move the clock. It is not saying that the people are healthier, fitter, or going to live longer. And it is a tautology, and it's something that is plaguing the aging field right now, because the methylation clocks are hypotheses. They don't agree with each other. I've recently seen data that they are largely reporting on inflammatory status. They're easily, changed by things like resolution of infection, and they do not necessarily predict longer-term health outcomes. So that's the early part of my talk in kind of busting longevity myths. So that's a little bit of level setting that I like to do when I come to a longevity conference, to get people to realize that there's a lot of hype out there. Now, what I want to tell you about from my own laboratory is work on NAD, because metabolism is, as far as I'm concerned, the vital force, the life force. And metabolism is not just making ATP, taking all the food that we eat and generating the ATP that runs of all of our cells and tissues. It's actually making and maintaining our whole bodies. And essentially, the 4 NAD coenzymes are NAD+, NADH, NADP+, and NADPH. And NADP and NADPH are largely involved in biosynthetic processes, in anabolic processes of making your whole body, as well as repair. And so now the thing that we found in 20 years of working on NAD and having developed a technology called quantitative targeted NAD metabolomics is we found that NAD comes under attack in many common conditions. So in alcohol use, liver NAD comes under attack. We've measured that in humans. Overnutrition. So if you overfeed a mouse and also drive it into a mouse model of type 2 diabetes, you attack the liver NADPH. Noise sufficient to produce hearing loss attacks the NAD system in the cochlea. Sun and oxygen damage, so DNA damage, attacks the NAD system by activating an enzyme called PARP1. Reactive oxygen species drives down the levels of NADPH in detoxification of free radicals. Irregular hours, time zone disruption disturbs the NAD system. We have some beautiful papers on pregnancy and postpartum as disturbing the NAD system. We and other groups have shown that heart failure in mice and in people disturbs the NAD system. We know that neurodegeneration, central neurodegeneration, and peripheral neurodegeneration disturb the NAD system. We show that coronavirus infection and inflammation disturb NAD, certain types of cancer, and then it's widely reported that aging in the NAD system declines in aging. It may be kind of the sum of all of these collective insults, or it may be something that is intrinsically programmed. We don't actually know. And so the idea of NR, which is depicted here on the right side, is that because NAD comes under attack in many of these conditions and metabolism is required for repair, and we know that metabolism declines in aging, that the use case for NR is to basically restore NAD in order to restore and maintain all of these vital processes and cellular health and resiliency. So that's the idea. Now, we started this project back in 2003, 2004 with discovery of the NR pathway, nicotinamide riboside. That's the biggest piece of NAD that can get into a cell. And we found, by the way, we found the genes involved in converting NR into NAD. They're called nicotinamide riboside kinase 1 and nicotinamide riboside kinase 2. We've done dozens of experiments in mice and rats. But as you know, NR has been commercialized by ChromaDex. I told you I consult for them. And so now there's a clinical trial, track record for NR. And I'll just summarize it here. First, safety. So we've done multiple safety trials, up to 2 grams of NR per day for up to 12 weeks. I think that there are much longer trials that have been completed now. We've never seen any serious adverse events that are attributable to NR. If you have enough participant days, someone will have a headache or a stomach ache, but you don't see an excess of this in the NR participants. You see basically the same types of events in the placebo. In one of the first completed trials, we saw lowered blood pressure in people that have moderately elevated blood pressure. In a trial for weight loss and insulin sensitization, NR did not really change weight loss but as a monotherapy. But when we looked at the participants, when we looked at the participants that had fatty liver, nearly all of them with NR had a decrease in fatty liver. And it was a big change from about 11% fat to about 9% fat. So a 2% absolute drop in fatty liver. That did not achieve statistical significance. Unfortunately, it wasn't randomized for fatty liver, and so it wasn't a perfect trial. However, with proper p-values and repetition in 3 or 4 different trials, Niagen depresses inflammatory markers like IL-6 and IL-10. That was initially shown in the paper that I was on in elderly men, but it's also been shown in people that are getting NR for heart failure. It's been shown in combination with 3 other over-the-counter molecules and to accelerate COVID recovery and with lower kind of cytokine storm, lower inflammatory markers. So the lowering inflammatory markers is a repeated result. We have a small trial that came out of Norway that showed improved cerebral blood flow in Parkinson's. I told you there's a positive result with COVID-19, and there's a positive signal in an ALS trial, and there's overall about 50 RCTs worldwide. So we're very pleased that this has been able to be translated, and there's a lot of results that are becoming available now. Now, what about NMN? I'm sure I'm going to be asked that question. Well, basically, it's known from the cell biology to act as NR. So NMN is NR with a phosphate on it, and compounds with phosphates don't get into cells. The phosphate basically has to come off. And there's much less human safety data with NMN. And in fact, because there's a company that is testing NMN as a drug, the United States Food and Drug Administration has sent out a letter saying that because NMN has been investigated as a drug before new dietary ingredient notifications applications were applied for, there appears to be a market removal in the United States. And so the use case for NMN was said to be Sirtuins, and you don't have to age, which I don't think that Sirtuins are important in human aging at all. And I don't think the use case for NR or NMN has anything to do with Sirtuins. Perhaps it's good that NAD has gotten better known because the story about NMN was so widely told. And as I said, the book has been extremely widely read in dozens of languages. Now, here's something that's kind of funny. There's a TV show here in the United States, I think it's based here in Southern California, called Keeping Up with the Kardashians. I'll show you a clip.
For the rest of my life, and I'm never going to age.
Okay, so I think that's Kourtney Kardashian, and
Just big on our health.
we're big on our health journey.
For a great friendship. I'm going to NAD for the rest of my life, and I'm never going to age.
Haley Bieber says, I'm going to NAD for the rest of my life, and I'm never going to age. Right, so what are the problems with this? There's no safe, she's taking an intravenous drip of NAD+, and there's no safety data for that mechanism of delivery. That mechanism of delivery doesn't make sense because NMN has 1 phosphate, NAD has 2 phosphates. And so you're putting NAD into the blood that's going to deliver NR. And while, that may actually, she had COVID apparently when she was doing this, while, that may accelerate COVID recovery by boosting cellular NAD by delivering NR, and that may allow you to age better and with more resiliency. We are, in fact, all going to age, and we shouldn't be telling ourselves these myths and hypes about NAD. So how to age better? I say choose your parents well. It's kind of a joke. Nature and nurture. So you get your genes from them. You also get your foundational habits of how active you are and what kind of food you eat. We know that human lifespan was doubled, in the last century, largely due to public health measures like clean water, vaccination, clean food. In the United States, I have to emphasize avoiding gun violence and drugs. Alcohol is not a help. Sleep is very important. You want to maintain high physical and mental activity with social engagement. I like to tell people that there's a lot of junky advice on the internet, and I think that some of the podcasts and the websites and the age kits that are targeted to the worried well are basically not helping anybody. They're robbing your time, which is your most precious resource. The age kits are a research tool. They don't have a value proposition to the individual. And I think that there is a use case for Niagen in building your resiliency and your repair. So I would like to conclude with a couple of take-home messages. First, I think we should exercise caution when describing breakthroughs. Remember 32 years and 9 months ago, we were told that aging was a solved problem and we can take senescent cells and reverse that and take living cells and make them senescent, and that 3 pharmas are going to work with us and we're going to solve all these different diseases and aging. That's just not true because there aren't monogenic longevity genes that would enable such easy approaches. So you have to understand the polygenic nature of longevity. I would say the highest tech longevity hack that I've ever seen are messenger RNA vaccines. And the use cases of nicotinamide riboside are varied because NAD comes under attack in so many conditions. And because the NR gene pathway, the nicotinamide riboside kinase genes, they get turned up transcriptionally when NAD comes under attack. So for example, in the heart failure model, NAD comes under attack. There's a bioenergetic crisis in the failing heart. AMP kinase is turned on, and that turns on the nicotinamide riboside kinase 2 gene, which makes NR protective for those mice. And that is the reason why there's NR clinical trials for human heart failure. And the clinical trial that I would like to see is wound healing. So if you were to compare, if you were to make little scratches or abrasions or very mild burns in, let's say, 25-year-old men and women and 65-year-old men and women, you would see the healing would take a longer time in older people. And what you would like to see is that you could improve the rate and the rapidity and the thoroughness of the healing process by boosting with a safe compound like nicotinamide riboside. I thank you very much for your attention, and I would be most happy to take your questions. I see Dr. Kapahi, great talk and effort to reduce the misinformation around Sirtuins. Thank you.
感谢 Brenner 教授的精彩演说。现场的观众朋友
Thank you very much for your wonderful talk. So now I'll check with the audience if we have any questions for you. First of all, thank you very much for the wonderful presentation. I have two questions about NR. Especially your introduction about Sirtuin. Now, I have two questions about the safety of it.
If Dr. Brenner is saying that Sirtuins are not longevity genes, then why is NR and NAD interesting? So I would like to emphasize NAD is the central catalyst of metabolism. NAD is incredibly important. NAD comes under attack in multiple conditions. Like we can't avoid the sun or oxygen, right? When we go outside in the sun, we get DNA damage that has to be repaired. When we function in oxygen, we generate reactive oxygen species. So those are inevitable parts of life, and both of those inevitable inputs into our life, sun and oxygen, just that simple, attack the NAD system. So the reason why NR is important is because NAD is important. Sirtuins are not the main mediators of NR. Sirtuins are not the main mediators of NMN. So NMN basically acts as NR. NR is important because NAD is important, and because as you go through life, your NAD system comes under attack. It's not going to make you live to 180 years, but it's going to allow you to age better. That's the basic idea.
No, actually, Sirtuin, I 100% support or agree with your genetic mutation cannot lead to longevity, this hypothesis. I totally agree with that. So I think my first question is about safety, because as you mentioned, the NR is very important in metabolism. And the most energy craving cell actually is cancer cell. So I know there's a lot of human safety tests, but in theoretical, is that possible that the NR can increase the cancer incidence?
Yeah, so that's a great question. I'm glad you asked. So, NR is part of a family of molecules called vitamin B3, right? So I discovered the vitamin activity of NR in 2004, but in 1938, nicotinic acid and nicotinamide were first described, right? And they were introduced into the human diet in the 1940s or 1950s. And nicotinamide is in all kinds of skin products. And there was a huge Australian trial to test whether oral nicotinamide would be preventative of skin cancer. And that had a positive result. So higher NAD status in a huge Australian trial was preventative of cancer development. Why? Because you go outside in the sun and there's DNA damage, and non-repaired DNA leads to cancer development. There are multiple animal systems in which nicotinamide and NR are cancer preventative. There was a recent paper about a month or two ago in which a group did kind of a crazy thing, and they took a human cancer cell line injected into the heart of a immunocompromised mouse, and they looked at the incidence of brain metastasis, and they came up with a very scary headline. But that's not a reasonable way to look at metastasis. That's not a reasonable metastasis model in the mouse. And in fact, just yesterday, I was shown human data showing that with thousands of human breast cancer specimens, that if you look at the survival rate of the women that had high expression of NAD pathway genes versus low expression of NAD pathway genes, higher expression of NAD pathway genes is a better prognosis in human beings. So I don't think that there is a legitimate scare for use of NR in terms of cancer incidence. I think it is almost certainly cancer preventative.
Okay, thank you. And I have the second question. Actually, it's about the mice model, because in your previous slide, you mentioned kind of misleading mice lifespan model. But how about the NR? Because I'm not quite familiar with NR. So how many mice model have been proved or been tested in NR about the lifespan?
I mean, in terms of all of metabolism, like we did a obesity, type 2 diabetes, and nerve damage in mice. We did protection against chemotherapeutic neuropathy in rats. We did heart failure in mice, but then we saw the same gene program in humans. People have done muscular dystrophy in mice. People have done mitochondrial diseases in mice. There have been there are probably 100 NR mouse papers. So there are, dozens and dozens of very positive trials in mice. And basically that has, that gives you an idea of where you want to do human clinical testing, right? Because if you see a positive result in a mouse, then you want to see if you can translate that. But you have to think about the human condition, right? Because you can induce overweight and type 2 diabetes in a mouse model in 2 or 3 months, and you can treat it in 2 or 3 months. But human obesity, may develop over decades, right? And so you have to do the human trial in a way in which you could detect a positive signal, and you have to randomize according to the primary endpoint that you want to measure.
Okay, thank you very much.
Yeah,
thank you.
我们现场是否还有其他的观众朋友想
Do we have another question from the floor? Hi, I'd like to ask that for the reduced form NR, compared with AR, what are the differences? And comparing with NR, does it work better? The reduced form of NR.
Oh, that's an interesting question. Yeah, so there's a way to basically NR with electrons is NRH, and it appears to be converted to NMNH through a different kinase, not nicotinamide riboside kinase, but adenosine kinase, as I recall. And so you can get extremely high levels of NAD, NADH from NRH, but we don't know whether that is entirely beneficial. So there's no human data on it. There's a little bit of mouse data coming out, but I don't know what the tissue distribution is going to be, because you're giving something orally, and then My concern would be that the reduced nucleosides and nucleotides like NADH breaks down in acid. So NR is stable in acid, just like NAD is stable in acid, can go into the stomach, and it should still be available. Whereas NRH, I would be concerned that will fall apart. But it's something I'm watching, and we have some new experiments to look at that.
So we may
safety data on NRH.
Thank you, Dr. Brennan. And we see that there's another audience who would like to ask a question. Let's pass to her the microphone. Hi. We have reported that aging is not a disease, but we can make it better or worse. But we cannot avoid aging. But I would like to say that you've seen many results of the longevity. Do you maintain that attitude? And also, what kind of clinical results will be able for you to reverse your attitude?
Aging's not a disease. Aging is a normal, genetically programmed developmental process that begins with fertilization and develops through birth and puberty and to reproductive prime. And then we have a loss of our capacity as we age, but no, I don't consider aging a disease. I think that with good lifestyle and potentially some interventions, we can have more people live healthy lives into their 90s and 100 and beyond. It appears that maximum human lifespan, is encoded in our DNA. It's probably around 120 years, and I'd love to see more people get to that age. But you can go from being like, I'm 61, for example. If someone were 61 years old and smoked and had a very sedentary lifestyle, and then spent the next 1 year getting off of smoking and eating better and exercising, they could go from an unhealthy 61-year-old to a healthier 62-year-old. They would be on an improved trajectory, and some of their biomarkers would have improved for the better. But I don't consider that an age reversal. Your age is your age, and your function is what is important. And so I think that the language that we use in this field is important, and I don't think you can credibly call aging a disease, and I don't think there's any rational basis for claiming age reversal based upon biomarkers. Of the biomarker thing. There were people that were saying they did biomarkers and they said, oh, COVID infection makes you 10 years older or something like that. I looked at those data and I said, that means that the aging clock that they're using is measuring inflammation. Sure enough, when the infections were resolved and they remeasured the people's so-called biological age, it reverted back to what it was. So you're not meaningfully talking about age when you're just reporting biomarkers. It's a misuse of the data.
Thank you, Dr. Brenner. Do we have any other questions? The man in black, Zeus. Hi, Dr. Brenner. I remember that last year there is a COVID research on NR. You know, now we have a outbreak of COVID for Omicron, this strain. Do you have any data on the clinical trials? And for the supplements of NAD, NR, and NAD, which one is better for the curing of COVID?
So we published a paper in the end of 2020 showing that coronavirus infection leads to an attack on the NAD system. So the mechanism is that double-stranded RNA, you know this is a double-stranded RNA virus, that hits an innate immune cell. It leads to an interferon response. And 5 of the interferon-induced genes are called PARP genes, members of the PARP superfamily, and they use up a lot of NAD. So coronavirus infection drops down the NAD levels in cells, and we showed in that paper that by providing NR, we can lower coronavirus replication. Based upon our preclinical study, there was a phase 2-3 clinical trial in Sweden and Turkey that showed NR plus 3 other compounds accelerated recovery of COVID. I think that NR was the active ingredient. It's being tested by other people, but NR alone, to improve people's function from long COVID, as well as kidney function and recovery and prevention of COVID. So if you wanted to try it, it would be using basically a therapeutic dose of NR. So in the United States, the kind of consumer dose is like 300 milligrams, and clinical trials that are testing NR are more like 1 gram, 1 to 2 grams per day.
问题我们请到
So next question is from this lady in pink. I'd like to ask that before about NMN is oncogenic. Some people say that. So NR is also like this. NR and NMN, and NAD plus, they might be oncogenic. So to a certain extent, precursor of NNA, will it also have this kind of disputes?
I don't think that the data are at all convincing about oncogenic. There's a hierarchy of evidence, right? You start at the top of the most convincing evidence are human data, right? And when you see that NAD precursors in large randomized trials are cancer preventative, that is much more convincing than an experiment where somebody does something in a tissue culture plate or takes a human, cancer cell line and injects it into immunocompromised mice. The higher quality data show that by enhancing DNA repair and enhancing immune function, that higher NAD status is cancer preventative. So I believe that the higher quality data show that NAD boosting with nicotinamide or NR or NMN, which basically acts as NR, will be cancer preventative.
Thank you so much for your support and brilliant lecture, Dr. Brenner. Now, I will extend my warmest welcome and respect to you and looking forward to your new researches in the future.
Okay, everybody come and visit me in Los Angeles, and I wish you very well for the rest of your conference. It's 7 p.m. here in California, and it's time for dinner. Thank you so much.
Thank you so much.